Okay, now the recording is started. So, the previous,
Ma'am,
yeah.
is there only structural difference between fibrillar and focal adhesion, or there any functional difference, ma'am?
So functionally, they are doing the same thing. They are, these contact points are helping the cell to migrate. Okay, the structural difference is there. Means what all proteins are coming and forming these contact points. That difference is there. So the previous 5 6 slides, the discussion for the previous 5 6 slides, I forgot to record. So if you guys have any queries regarding any of the slides, I will explain to you guys again, so that in future you can have a look at that recording. Okay, so now the recording has started. So Murli, is your query clear?
Yes, ma'am.
Yeah. Okay. So we have now already uh, saw three types of integrin based cell junctions, cell ECM junctions. Now it is important to note for us that cellular responses are different. When you are observing the cell response on a flat surface, on a planar surface, 2D planar surface, in comparison to 3D hydrogens. So when cells are seeded on a flat surface, then the response will be different, and when the cells are encapsulated inside 3D hydrogens, then the response will be different. So one should consider the biochemical composition, means what is the composition of the material that you are using, the mechanical properties, and as well as the orientation of the ECM or whatever material that you're using, how the uh, polymer chains are oriented, etc. So you guys should think about them before designing any tissue engineered scaffold, because the response will be different. As you have seen, different types of integrin based junctions are formed by the cell. So depending on what kind of substrate is there available to the cell will form that kind of a junction. Now integrins can also bind to small peptide fragments within, actually binds to small peptide fragments within larger ECM molecules. So you guys have already seen the figure of fibronectin, like initially I showed you. But there are small binding sites for different type of molecule. One is the binding site for the integrin. So if when people have sequenced the sequence of the protein like fibronectin, they found what is the amino acid sequence of that particular integrin binding site, and based on that information, they synthesized small peptides like tripeptides or more than 4 5 amino acid like peptides. So it's very commonly used peptide, a small peptide, is the RGD peptide. It's a tripeptide composed of arginine, glutamine, and glutamic acid and glycine. So these three, RGD. This is the one letter abbreviation of the amino acid names. So RGD peptide is very commonly used by the tissue engineers to modify the surface of the scaffold, especially the surface of the synthetic scaffolds, where any cell uh, which cannot promote cell adhesion on its own. So they conjugate these small tripeptides to the surface of the synthetic scaffold, and then only they can see some cell adhesions. Okay, so they are basically trying to mimic the ECM in their artificially synthesized scaffolds. So, but if we will see the difference in materials, different type of materials derived from different type of source, and how they uh, change the designing of the scaffold for you. Okay, so there also we will come into this kind of peptides like RGD peptides. Now most ECM molecules, they have a number more than one integrin binding domain. So there is not only one, but there can be multiple integrin binding domains. And in some cases, binding of more than one peptide presented in a specific conformation is required to elicit an integrin mediated signal. For example, presentation of both RGD peptide and PH SRN peptide. These two peptides, they has to be presented simultaneously in order to engage this integrin, alpha 5 beta 1 integrin, and then only integrin mediated signaling that is going to happen inside the cell, that will get switched on. Okay, and the cell will show some appropriate response. Okay. Now, how many binding sites are needed to generate a cell response? Okay. So the RGD tripeptide binding sequence, they have, like, people have immobilized them, means but through covalent bonds or through hydrogen bonds, etc. People basically conjugate this RGD peptide with the polymer chain, synthetic polymer chains. Okay, and make the surface, modify the surface in such a way that the cells can grow on it. And people have done it at various densities, like per centimeter square there can be 10 RGD peptides or 100 RGD peptides, and they have studied their effect and tried to understand how many such cell binding sites are needed to generate a cell response. Cell attachment, spreading, and growth, all three were examined as a function of surface density of the RGD binding sites for the fibroblast cell. So one cell, for one type of cell, they have done this study. So the results what they have obtained, uh, that tells us that the average receptor spacing of 440 nanometer was sufficient for cell attachment, okay, and spreading, and 160 nanometer for focal point adhesion formation. Okay, so what these statements tell us, that the surface density of the RGD peptide should be such that there should be average distance between two RGD peptide of 440 nanometer. If that is there, then only the cell will properly adhere and spread. So first thing, whenever we seed some cells from a cell suspension, then first thing that the cell does is adheres and spreads, or tries to takes up its morphology. Okay, once that is done, then only it will be able to respond to a particular signal. So here they are trying to maybe cause cell migration. Okay, so for the cells to migrate, the cell will form the focal adhesion points. So for the formation of the focal adhesion points, they report that the spacing, average spacing between between two RGT peptides should be 160 nanometer. So this is the average distance, and from that you can understand what could be the surface density, means per centimeter square how much will be necessary, correct? So that this is in terms of length, and you have to just uh, calculate or correlate with the area. Okay, now how do cells adapt to surfaces in flow field? So in what kind of scenario can you expect a flow field to take place? Like, there's a flow of a fluid happening and the cells has to adhere under this kind of a flow field. When can you expect that? Anyone? Am I audible?
Yes,
Yes,
ma'am, you're
ma'am.
audible.
Yeah. So did you guys understand my question?
Ma'am, no, ma'am.
So I'm asking, inside our body, when can you expect that the cells has to adhere in presence of a flowing, circulating fluid? Where that kind of scenario will happen inside our body? Understood my question?
No, ma'am. You're asking when will a cell try to adhere in a flowing condition?
Yes, yes. So where that does it happen? Where in our body?
That's circulation.
Correct, correct. So yeah, so the blood cells which are there, okay, the macrophages and different type of immune cells like macrophages or the neutrophils, etc. So they are having the adhering capacity. They can adhere, okay. So, but they have to adhere while in a continuous flow system. So blood is continuously flowing in the body at different pressures, and under that pressure condition they has to adhere. So that is the question, how do they adhere then? Okay, so cell adhesion is a vital component in the response of immune cells in the circulatory system to infection, as they attach to the endothelial surface to then migrate to the target site. So through the endothelium layer, endothelium lining, what they do, and that is the surface on which they adhere. First, the endothelium lining and the endothelium then help them to migrate to the site where there is some infection going on, where they have been summoned by the resident cells to tackle with the foreign agent. The process of addition in this kind of a case is the result of a balance among the bond lengths, then the dissociation rates, and the affinity strengths. So these three terms, bond lengths, dissociation rates, and affinity strengths, I hope you guys will be able to understand now, because we have already seen some expressions where we have discussed what is dissociation rate, etc. So as the cell rolls along the adhesive surface, the balance of these forces means the forces governing the cell receptor affinity, then cell receptor dissociation constant, okay, then the what is the bond length between the cell, that the bond length for the bond that is formed between the cell and the ECM. So these things, there should be a balance between these forces, then only adhesion will occur in this kind of a flowing liquid condition. Now, how the modification in the ECM happens. So we will understand it through an example. Cells produce and degrade the ECM in their own environment, leading to a feedback loop on the cell fate and function. So I already told you that there is a feedback mechanism going on in the tissue. The cells synthesize and degrade the ECM component. Basically, they are continuously modifying the ECM structure, whereas the ECM has the molecules or the sites through which it can direct the cell function. Okay, the ECM molecules, they are themselves quite large, and because of which they have a very low diffusivity. Means they do not diffuse at a very high rate in comparison to the soluble signals. As a result, the time scales for ECM remodeling around the cell, they are typically quite long. So obviously, it's a big, large protein molecule. The sequence will be quite large, and usually you can imagine that ECM molecules, they are like collagen, they are like fibers, then fibronectin, etc. They are like globular proteins, or they can have subunits, okay, and forming one large protein. So this kind of structures is there for this kind of ECM molecules. So the sequence, protein sequence for these ECM molecules, they are also quite large. So it will take a longer time for synthesis and then post translation, assembly and all. So the synthesis of the ECM molecule and ultimately their positioning in the ECM, that will going to take a large time. And the length scale means to what extent or to what area around the cell the ECM is modified, that is also quite small. means at a particular time for a particular cell only few ECM molecules can be modified. Okay, so length in the area or the distance to which the ECM modification is possible at a time is small. So take an example for the cartilage tissue, where the resident cells are the chondrocytes, and the cartilage tissue majorly have the ECM component collagen. Okay, so collagen is a major component of the ECM of cartilage. So the production of the cross-linked collagen, it occurs in cartilage on the order of days. So it takes days, not few minutes or few hours. It takes days. With the penetration into the surrounding ECM of approximately 100 micron per day. So you see, this it takes a long time, like few days, and also the length scale is also quite small. Means slowly in one day only 100 micron of the distance. For the 100 micron of the distance, the collagen is secreted, and then again another day another 100 micron. So tissue is very big. Okay, but the modification is taking very slow time. So remodeling phase in the wound healing process that we have already discussed, that takes years. Okay, so as a load bearing tissue, cartilage is a load bearing tissue, and it is composed of an ECM that is constantly being synthesized, degraded and remodeled in response to mechanical deformation. So हम लोग को कभी चोट वगैरह लग जाते हैं या फिर anything heavy falls on our limbs etc. where the cartilage is there, then cartilage tissue is damaged. So in order to deal with that damage, the cells are continuously synthesizing ECM molecules or removing the old ones which are damaged in order to maintain a required mechanical property of that particular tissue. So people have evaluated, okay, evaluated how the ECM modification takes place around a single chondrocyte cell. So what their results show that there is a rapid degree of proteoglycan deposition in the ECM that is happening immediately surrounding the chondrocyte. So proteoglycan is another ECM component found in the cartilage tissue, and the rate of synthesis of this proteoglycan is quite rapid. Okay, it is happening quite fast. Okay, the deposition of the new ECM, it always occurs in the direction perpendicular to that of the compression. So suppose some kind of injury has happened, okay, on the cartilage, and just perpendicular to that direction, okay, the proteoglycan is synthesized and deposited. Why do you think it will happen? Can anyone imagine the situation where the ECM component is getting deposited, okay, at a site in such a manner that the say any force is coming, a hammer has like mistakenly fallen on your limb, like hand or your leg. Okay, so where cartilage tissue is there, so from top the hammer has fallen, so just below it the perpendicular surface is the bottom surface. So at that particular point the cell that is there in the cartilage, it will try to synthesize ECM component like proteoglycan in order to cover up or recover the mechanical strength of that particular area. Obviously some damage will happen, and immediately in order to overcome that damage the ECM component proteoglycan is synthesized. Okay, now these results typify the degree of physical control that the cells exhibit on their extracellular environment. So the cell has a very strong control over its ECM environment. Physical loads lead to direct cellular responses and thus modifying the ECM in response to particular conditions. So here some kind of load etc. has fallen on the cartilage, so immediately cellular response is to repair that particular area by synthesizing the ECM. So the ECM is getting modified. So that's it from my end for today's class. So if you guys have any queries related to the topics that we have discussed last week and as well as yesterday. You guys can uh, discuss. You can, you can ask me now. I can take it up. So tomorrow there will be a quiz of 1 hour, okay, in the evening time, 7 to 8. So today, actually, I had an experiment going on, so because of which I had to postpone the class till 8 p.m. Tomorrow it won't be a problem, so our quiz will start from 7 p.m. only. Okay. So any queries?
Ma'am,
Yeah.
from where are you taking those numericals for this, ma'am? From any book or from any articles?
So yeah, I have shared with you the first course handout of this course, right? Remember in the beginning? So there I have mentioned the name of the books that I am following. So you can check that document. The book Tissue Engineering, that book is there by Sangeeta Bhatia and Palson, Bernard Palson, as well as there is another book where it is titled Principles of Tissue Engineering by Bacanti, Joseph Bacanti, I think. Okay, that book is also there. These two books are being used for, for covering this course. That is already mentioned in that particular document. If
Okay,
you
ma'am.
anything else, no queries. All the topics are understandable to everyone. Okay. So let me then just take attendance.